Oops, there.
I'm sorry, I always forget to record.
Okay. So, when we talk about the disease triangle, the first thing, to understand is each plant has a set of diseases that could be a problem on that plant species. So the three diseases we're talking about here today, each one is only really specific to that crop. Well, Fusarium hub blight will go to corn as well. But it's important to understand, what crops are susceptible. And we can also look at using variety and hybrid selection to reduce that risk to that plant. And then it's also important to understand when the disease may move in, what growth stage is most susceptible to infection. And that's going to play a role in whether we have a problem for that disease in the season. And then the other thing to think about is where is the pathogen? So all three of these diseases, they do overwinter in each of the fields that, could overwinter in your field. So it's important to understand what is the field history of your crops. Do you have a problem with these different pathogens? If you do, then we look at what tactics can we try to do to try to manage those diseases within that system. Some of our diseases don't survive in the Midwest or in our fields. An example that I could bring up is, southern corn rust. We watch that spore movement in every year. So we start with a clean slate. We just monitor some of those diseases to see if they move in. But when we talk about Fusarium head blight, it's overwintering in our corn and wheat debris. And so that pathogen is already present in a field. So you have to understand the history of that field. Has it been a problem in previous years? Tar spot, we've now learned, it does overwinter here in the Midwest, but that pathogen also is moving on spores and weather systems. So not only is it field by field issue, but it can be a problem as we have a spore release and the movement of those spores across the Midwest. And then white mold being a soil-borne pathogen, if people have white mold in your fields, you kind of know that that's going to be a field that we have to worry about that disease. And so then the other thing to think about is the environment. So that's the dictator of what's going to be the risk for that season. We're going to share some of the tools we have available to look at those environmental conditions to maybe predict whether that disease is going to be favorable for each season. Each disease has a set of temperature, moisture, and leaf wetness requirements. So if we're not favorable, then we have low risk for disease. Maybe we don't need to implement additional tactics for that season. If we've had a continual problem, we have that favorable environment, that's where the disease ramps up. I miss anything, Damon?
Nope, I think you got it covered. I think the biggest thing, though, is, paying attention to this environmental component. I think it's safe to assume, at least with the three diseases we're going to talk about today, we pretty much have resident inoculum, and, we've got lots of corn and soybeans across the state, as well as small grains in the Midwest and, various areas of the U.S. here. And so I think the environment is really that piece that we got to pay attention to and hone in on, in my opinion.
Okay, exactly. And so then if we look at general disease management in field crops and even for organic grains, I think of four regions. In grains, you have some change, organically, we have some different changes that occur than our standard conventional corn, soybean rotations. But the first and foremost, again, if we can identify genetic resistance to that disease, which I think is really important in organic production, then we limit the risk for the future issues that may appear if we have favorable conditions. It may not eliminate it completely, but again, we're hedging our bets to try to protect against those certain diseases. And again, that's the host side of the triangle that we're acting on. We can look at crop rotation, and I think in organic systems, we do have an opportunity to incorporate more different, a range of different plant species within that rotation that may help limit the survivability of the different pathogens. So if you think about it, if there's not a host for that pathogen that's surviving in the debris, then there's more opportunity for that pathogen, the propagules that are going to spread, to be reduced in that field and maybe reduce that initial development of the disease down the line in the future. But again, it's going to depend very disease specific, whether the rotation will favor or disfavor that pathogen within that system. Sometimes the other thing to think about on the sanitation side is, we can plow pathogens under. We can plow that residue that they're surviving under and expose those pathogen propagules to other organisms in the soil that may help, again, reduce that initial inoculum that could lead to the disease developing in that season. And then I think it's also important to understand avoiding the spread of inoculum, particularly with white mold is one example, where if we have a field that has a bad history of white mold, we don't want to be moving those sclerotia in the soil that are surviving to a new field and then causing a problem in another field that didn't have the previous problem of that. And then finally making a decision on a fungicide. Now, this is where we kind of shift gears versus conventional versus organic systems. We have a lot of fungicides. We've looked at a lot of fungicide efficacy in our conventional systems. And that's where we see there's a gap in understanding some of these products that may be able to help reduce disease in the organic system. And so that's kind of some of the work we've been trying to look at, is pool some of the products that may be out there that are labeled to maybe try to manage some of these diseases and really look at, are they efficacious to control these diseases in grain crops? There's a lot more work that's been done in vegetables and fruit crops. I think that's where we're lacking, is looking at some of these products. Can they be applied in controlling some of our pathogens within the grain crops? And so I don't have any, I was going to move on to just start jumping into some of those pathogens, unless anybody has any initial questions on what we've discovered right now or talked about.
There is a question in the chat. And I don't know if you're familiar with Rick Clark Darcy. He's a farmer from Indiana that's been quite on the speaking circuit over the past year or two. But he's claiming that on his farm, extensive cover cropping and building soil health has minimized plant and disease pest and disease pressure. Have either of you seen this happen or can speak to the role of either cover crops or soil health on incidence of disease?
Well, I mean, people have been working on suppressive soils and things for, decades now. Darcy and I learned this 20 years ago, and we took plant pathology. I think one of the things that we've seen in modern agriculture, and especially in the conventional systems, is a lack of rotation for one thing. So, if you look at just corn, for instance, we have a lot of corn on corn. That reduces diversity, right, of your other microorganisms. So I think there's something to be said about a diverse rotation. There's something to be said, I think, about cover crops there. Folks are looking at those diverse rotations and things, and looking at the microbial diversity, and in general, it doesn't hold for all pathogens, but in general, and I think Darcy can comment on this too, pathogens can be sort of weak competitors, right? So there is that. I think the other big piece that we have to look to is that in modern breeding programs, we're pushing so hard for yield that we don't preserve genetic resistance. And I think this is another piece that has to be considered here, because some of these pathogens are going to still exist whether we have good microbial diversity or not. And so then it comes down to utilizing, say, genetic resistance to, give us a good baseline level of management. And so I think the breeding needs to, sort of slow down on the yield side of things, in my opinion, and try to bring in some of that genetic resistance into the breeding programs.
Yeah, I think you answered it, Damon. I think, we can look at many of the diseases and we can improve the ability for those diseases not to be a problem. But then we got to just keep in mind, there's some diseases that you may do everything perfect in your field and it's going to blow in from the neighbor. And I'm going to, here in Indiana, dealing with tar spot, I think if you haven't seen it, then I don't, just be aware of this disease, but there may be some diseases that we have to be cognizant of. They could blow in even if the field health is great for that pathogen. So the soil-borne diseases and maybe the fusariums and the white molds that are surviving, and we can break those down, that we can reduce the risk. But then there's some other things where if we get bad years like we had this last year, and some of the other diseases that are moving around in weather systems, we're not going to be controlling it as much. So it's a balancing act. And I think it's important to understand what is the risk. And if you do have those risks, and maybe you don't see many diseases on your farm because you've improved the soil health. And so then you don't have to worry about it. But I do think it's important to get out there and look. And I think there are other things that could pop up. And then, we get the call at the end of the season when there's a breakout of something. And so that's just something, to keep in mind. Or if we know that it's going to be a bad year for some of these diseases that may blow in on the wind, then maybe there's some opportunities for other things to try to protect the crops. So that's kind of. And so the first disease we were going to review, I lost my screen here. We'll start with wheat. And so we were talking, Fusarium head blight, and I think we've gotten lots of reports with issues in organic grains of some years with the issue with Fusarium head blight. So this disease does overwinter in our debris, and it, overwinters in wheat and corn. And so we can, we get it's the same pathogen that causes the stalk rots and ear molds in corn, and then can, cause issues with head blight in wheat. And so we do monitor this disease. It's, survives the debris, rain and wind move the spores up into the crop canopy. So on wheat, it's infecting at the flowering stage. Just as we're beginning to flower is when the infection occurs, and then 7 days after that, and then when we do have infection by the end of the season, you can see these kernels that become blighted, and they may be infected with the pink pathogen there. You can kind of see that in the center of that head, and then we may have some issues that down the line lead to the development of DON or vomitoxin within that grain. So that's some things important to understand for Fusarium head blight. So again, a lot of work on wheat production has been done on trying to protect that head from infection. So we've shown that early anthesis that's when it's really getting in there, and we need to protect the head. So there's a lot of tools that are available to help us predict when the pathogen is going to be active, the conditions are conducive for those spores to be moving into the wheat, especially if we're at that early anthesis under those conditions. Then we can try to make a prediction if, it might be important to put out a protectant fungicide or organic product there and trying to manage that disease. So there is a tool that's available, and they've just updated this as of this week, and the new system will be going live. I just pulled some examples just to show what it provides. So this is a Fusarium risk tool, and this is looking at 2020, and you can select. So part of the tool is looking at susceptible varieties versus moderately resistant or moderately tolerant varieties. And so if you look at this risk map, so on this map, the oranger areas are a higher risk for infection to occur if we're at that early flowering stage. And so that's what we monitor is the weather conditions conducive for infection. So in a susceptible variety, that range of development can, is wider than if we had a moderately resistant variety. So that's looking at the middle of May. Then we look at a moderate resistant variety, less risk because we've deployed resistance within to those varieties that we selected. And then we can move in further in the season. So if we look down at the end of May, and so if we had delayed planting or a variety that's delayed and flowering, maybe as we move up north in Wisconsin, you can see the weather conditions shifted, and now we have a greater risk across the whole country for this disease. It may, a tool to make a decision if we need to put something out to protect it. Again, looking at moderate resistance, it contracts back a little bit because we have that resistance deployed to try to minimize the disease. And so some of the work we've been working on with this new project is we wanted to look at 2 different varieties by some of these newer products that are available, and do they provide the protection compared to our standard conventional product. Procero is what we used. And so we had 2 varieties planted in our field trials here in Indiana and in Wisconsin. And then we looked at some other products. So we had Champion, which is a copper-based organic product, Pacesetter, which is a Rhyonitra extract of knotweed that is used in organic production, Sonata, which is a bacillus bacteria that can protect the leaf. And then we also looked at Actinovate. And so what we did is trying to make these selections is try to look at those products that might be labeled for that crop, and then also just utilize where we've seen maybe some activity in other crops where they've been beneficial in trying to control other crops in organic production. And so what we looked at here is just looking at this integrated tool management. So looking first at the hybrids or the varieties. And that's where we really saw some differences this past season. I would say the risk for scab this past year was low. So we didn't have the conducive conditions, at least in Indiana, for risk, but this is looking at those 2 varieties. So you can see, in Wisconsin, actually, Damon did see a significant difference between, Fusarium head blight is the measurement of the infection of those heads. You can see that the Harpoon did provide some better resistance to infection. They also saw a boost of yield there in Wisconsin, and then I can't see, and a reduction in DON, which is the ultimate goal too, as well as we want to reduce that DON level if we can, where sometimes we might get dinged if scab gets into our varieties. But combining across locations, again, we did see that Harpoon yielded better, and then also that we could reduce DON with the good variety selection. So that's the first thing I want to hone in on in organic disease management. I think variety selection is key. And it's important to try to look at some of these varieties to try to identify those that may reduce the risk for us in the future.
Yeah, Darcy, I think there's a lot of opportunities here actually in the small grain side of things. One thing about small grains breeding is it's still in the public sector. And so it's all, these are all non-GMO conventionally bred varieties. And I've looked at Kaskaskia and Harpoon over many years now here in Wisconsin and comparing the two. And we've had years where we've had really significant pressure of Fusarium head blight in some of our conventional trials, and Harpoon is so resistant that it really doesn't need to be sprayed in most years, believe it or not. So, my goal, and how I think about this is if I don't have to make a fungicide application at all, that's a leg up for me. It's one less decision I got to make. It's one less product I have to get out in the field. And so I think there's a lot of opportunity here in the small grains to do some homework. The other thing I'll mention is that we have run into issues with uneven head emergence here in Wisconsin. And so that's also something that as you evaluate a variety, look at that, because we're dealing with a polycyclic disease, meaning there's several cycles. We can have several spore flights over a period of time, as long as that weather is conducive. And so in some years when we have these, secondary tillers or even the primary tillers, depending on weather issues, that are delayed behind some other primary tillers, trying to make some of these decisions to get a fungicide out, whether it be organic or conventional, can really be tough in making that decision. And so you can take some of the guesswork out by just looking at head emergence and evenness in the field and trying to look for varieties that really fit your location in addition to the resistance level.
And so in Indiana, we did have a little bit of separation on products, mainly, I think I don't have the letters in there, but we saw some activity, I guess, with Champion being close to Fusarium. But again, our disease level was so low that we really didn't see any good significant separation. But I think there's some products that may be tending towards giving us a little more protection for head blight moving forward. It's just they're designed a little different. So looking at a copper-based product, it's going to wash off. It's more protectant. So that's important to understand, as we got to get it out there in a timely manner. I don't know what else I had. And so we did see differences in the damaged kernels, so the Fusarium damaged kernels, but mainly, the Prosaro was, our standard fungicide. But I acknowledge, looking here at Actinovate is one product that jumped at us at protecting some of those damaged kernels. So possibly another product. And that's the one product I've seen in all three trials where we're seeing some additional activity. It is a streptomyces, so it's a bacterium that's colonizing and trying to inhibit some of that fungal infection. So just something to think about if you're looking at maybe having a backup plan. And so that's what I look at, looking at organically. These are the backup plans to, if you do everything right, and then you get into a season where we have such conducive conditions for scab, maybe there's something here where we can get a little bit more control by adding a biological product that is labeled for organic production. That's kind of what we're aiming at.
Yeah, it's important too to consider, you mentioned the washing, Darcy, of Champion, and that's, you need to be careful of that, because when we're looking at these, say in a controlled greenhouse setting or growth chamber setting, some of these products look really, really good. And then we carry them to the field, and that's where things get challenging. Obviously, the populations of other microorganisms are at play there, but a lot of times, especially during the spring and early summer when we're treating for Fusarium head blight, it's rain washing. And so products like Champion now, there's a challenge there in just trying to keep the product actually adhered to the plant. So that's something to consider as you look at data and you're making decisions on which products you might go after.
And I guess I would say that's the biggest gap that I look at when I look at biological products to begin with, is that a lot of the data is coming out of the greenhouse and growth chambers. And when we take it to the field, we do not see those expectations. And I think there's two layers of things that we need to think about. First, they're not the silver bullets of our fungicides that, we're used to seeing on the conventional side. And secondary, what are we doing within that system, especially if we're introducing a new living organism? we have to be cognizant of where it needs to be and then what else it's interacting with within each field. And I think sometimes it'll be a field by field situation of whether those new biologicals will actually have some activity within that system.
I agree.
Okay. And so looking at head blight, generally, and we're going to keep pushing a hound on this. I think that'll be the theme is that, variety selection is going to be key in most organic systems for disease management. Finding those varieties that may have a little disease protection, making sure we have high quality seed. Let's start from that end and then look at, if we can avoid some of these cultural practices, if we can rotate correctly, if we can avoid water management, maybe we can limit the risk of Fusarium head blight. And then down the line, if we still look like it's going to be a bad year for it, then, hopefully in a couple of years, we'll have some data that will show some products that might be an option to put in there that would give you a little more protection. Then I don't think we'll find something that's going to be the silver bullet, but I think it's layering these in and reducing the risk. And I think that's what we're going to see for all these diseases. Also, we just want to note, if we do see we have a bad year of Fusarium head blight, it's important we can adjust those combine settings. Work that's been done up I think in Wisconsin is, gives you that, changing the fan speeds and blowing out some of that poorly grown seed out the back of the combine will help reduce some of that risk for DON and vomitoxin in the harvested grain. So that's just something to consider. If you start hearing words from Damon or I that we're having a bad head scab year, then we need to think about how we're going to harvest those fields as well, just to try to reduce our risk for the DON production. Now I'm going to turn to tar spot, which has been on my circuit for the last 6 months. So I would say this is like my 60th talk on tar spot. But anyhow, today it's a little different. I like talking about some other diseases. So it's kind of fun to insert them. But just as a reminder, in tar spot has been a problem in northern Indiana since our 2018 epidemic. Still, I'm mostly worried in Indiana at the northern part of the state. And if you haven't had this disease on your farm, I don't think you need to worry about it, but just be aware of it. That's my message on that. If you haven't seen it, just be aware that it's around and it is causing some significant problems for our growers around Lake Michigan, where we have the lake effect weather. So this new disease, yep.
Right, before you go on, there is a question in the chat about the benefits of surfactants in field conditions.
Yeah, I think possibility, especially spreader sticker type adjuvants that can get us, better adhesion. And I think, we're sort of thinking along those lines. The trouble is trying to find good OMRI approved ones, right, that we can pair with some of these. And so I think that is certainly something to look at, because again, adhesion is one of the biggest issues I see, especially in our environment where it rains all the time. So I think, yeah, those are places to look. And I think we're all kind of thinking along those lines. I don't know, Darcy, if you've looked at any of those at all to see if they've improved.
Not yet. There might be some newer ones coming out that are orange extract and things. I've seen some of those, but we're still just getting into that. So how do they interact with the compounds that are labeled organically? And then, so I'm also going to say, if we're going to look at a living organism, we have to be very careful what we're tank mixing, because there are some of the products can be mixed with Champion and to get fine, but you got to look at those labels to see if the company's even done that kind of work, that we don't want to be mixing something and then killing that organism. So that's some of the questions we have, and we just haven't looked at all that information to provide that data. But that is something down the line we can add into our to-do list. Okay. So yeah, getting good leaf contact, particularly I would say with tar spot as well, is going to be critical in trying to protect against this disease. So we do know that it overwinters in the residue, and we've shown this working with Damon, that those spores are at least viable for 1 year. Now, I don't have the answer how many years down the road will this pathogen survive. I do know I've been in fields here in northern Indiana that were full tillage, soybeans, there was very little residue, and it had to have been in the corn in the previous season. So I know we're at least getting some survivability in multiple years. That being said, in addition to just surviving on the residue, once this disease is ramped up in our region, we're spreading those spores through weather systems. And so you may do everything optimum in your field, but those spores may blow in across the neighbor or from the next state over, and they're going to land there. And depending on our season, could lead to significant losses, particularly in the regions where we've seen tar spot now multiple years. We do know that those ascospores can be released. And so fields with a history, we're seeing the disease move up from the bottom of the crop canopy. We generally takes about 14 to 21 days for us to see these stromata, these black spots that first appear. When I'm talking to my growers, when we see those spots, though, that's the end of that first life cycle of the pathogen. We're now the spore structure, that's the black stromata. It's then getting ready to release more spores to have the cycle repeat under conducive conditions in the crop canopy. And the thing we're seeing in Indiana is once this thing gets roaring, I can go from green canopy to a blighted canopy in a couple weeks, particularly as we're approaching early dent stages. This really ramps up and it can cause some significant issues with yield. And then we also have some issues with lodging risk as well at the end of the season. And so this disease is just a new disease. If you haven't seen it, I just would, want you to remind you to be aware of it. Right now, I still think our hot spot is around the Lake Michigan area, but I know we continue to see that area spread. In Indiana, I've got pockets now in Southwest Indiana where they're along a riverbed. And so we talk about environment dictating this diseases. This disease really is going to be dictated on how conducive the environment is.
Yeah, and I think the other thing to consider too here, Darcy, is that the nature of this disease, whatever we try to do from a management standpoint. So as we start to dig into, a discussion on management here, bear in mind that we're just delaying the inevitable. So we, when it comes to disease management, we have to lower our expectations, unfortunately. Right. And, this disease is one of those, right? Because, you can do everything right and the plants are still going to succumb to tar spot in a conducive year. So that's something to bear in mind. Just because you see it and you have an epidemic doesn't mean you're doing things wrong. It's just, did you slow it down long enough so the plants could beat the fungus? That's our strategy here. So keep that in mind, in my opinion, as we talk about these management strategies.
Right. And I guess that's the other thing is I don't say disease control anymore. I think it's managing. We're not controlling this one at this point in time.
There's a question of whether varietal resistance is rated by anyone besides the seed companies.
Yeah, I mean, we're all taking a look at varietal resistance and things. I would say that it varies by crop. So again, if you look at small grains, those ratings are quite accurate and consistent across programs because that's a public breeding effort still. So all that work's being done at the universities, and there's a coordinated effort in terms of the rating systems. When you move into corn and soybeans, that varies now. So, all the companies have a different rating system. The universities, when Darcy talks about tar spot here, we've got a coordinated system that we use to rate. So at least you can compare those across the university ratings. It is a challenge, and I know that some of the seed companies are working on trying to get better ratings out, but I think just hone in on what the universities are showing you in terms of what's going on in some of the local variety trials or hybrid trials and that sort of thing.
Yeah, I'd say we're limited, and I also tell my guys, look at your corn, look at your own varieties on your farm and see how they reacted, because that's you can actually see differences. And so we do see there is a range of susceptibility. I don't share that slide with us today, but there is a range. But again, everything can be infected to one level or another. And some of those that they're just holding back the disease to a certain point, letting that crop get to its natural maturity. I know the companies are trying. It's just hard to get in the right locations to get that disease data, and they want to see it multiple. We like to see things. We're sharing 1 year of data, and really we want 2 or 3 years of data. Really hone in on how active, how good these products might be in controlling the diseases in various environments. But if you haven't seen tar spot, so this is this example here on the left, this green leaf, that's the stromata that we talk about. That's where we get its name. It looks like someone splattered those leaves with black tar. It's the actual fungal fruiting body within that leaf. You can scratch at it. It's embedded and growing within that tissue. At this stage, this is what I talk to my growers. I go out here, if this leaf is up in the middle canopy or already at our ear leaf, and I have 5 to 7% infection like I see here, I can come back 2 weeks later and have the leaf on the right. And that's the thing that's really concerning with some of the growers I have, is that it changes so rapidly once it's going. And so we really need to get ahead of this. So we need to be managing it to slow it down. We need to be ahead of that when we're trying to time fungicides, and look at trying to incorporate other management tools within that. And so that in the center picture, you can see the rapid blighting that occurs as we continue. That leaves are just consumed by this pathogen. And so I've been in fields this last season. We had another severe epidemic. We were at dough stage in some of those fields and all the leaves were brown. So there was really nothing left to push that corn through to the end of the season. And so we did get out, and we do have a good site where we're trying to evaluate some of those organic products. And so in corn here, at least we started this past year with only one hybrid or variety. We're going to be looking at 2, hopefully this season, to see if we can integrate that. But again, we went with our standard. What we found with Headline Amp, the VTR1 has been working relatively good at providing protection. Now, again, these are, I want to say there's limitations in even our fungicides. They all have a window of activity. So when I talk, we talk about putting out our fungicides, they're active from 2 to 3 weeks, and then they're gone. And this disease is one disease that's going to ramp right back up again if there's green tissue. So that's important to understand, is we're trying to time it, put a well-timed application out to try to control it on the conventional side. And so then we've also looked at some of these products. Can any of these products, slow the pathogen down enough to provide a little more protection on that yield loss that we might see? And so in Indiana, we've experienced up to 50% yield loss in some of our fields this last season. So that's the concern, is can we even, can we protect some of that yield? On the conventional side, I see generally 12 to 40 bushel protection with our fungicides, depending on that season and how early the disease started. And so looking at these products, again, we have the copper, there's Oxidate out there. Oxidate is designed more to be like a spore killing application. So it really only has a 4-hour activity window, but maybe we've knocked the spores down enough a little bit to get a little more protection in there. Actinovate, again, is that Streptomyces, a bacterium. Serafel is a Bacillus amnioefficacious that also provides protection. So we were trying to see if any of these, if they're out there, can inhibit that infection period and slow down the disease. And so looking at Indiana here, actually both Indiana, Wisconsin. So I colored Wisconsin red, Indiana's gold, yellow. So again, our Headline Amp is our standard fungicide. We can get good protection from tar spot in both locations. But I did have a couple products in Indiana that seemed to reduce the disease slightly. So the Serafel had a little activity, Actinovate had a little activity, Badge is the copper, that's a protectant again, did provide activity there. So actually we saw some activity in all the products in reducing disease slightly. I don't think we saw, there was much change in yield. The Headline Amp and the Badge were the 2 that really increased. My letters are backwards. I look at it. So the C's should be A's, but yeah, so I think the Headline Amp and Badge did see a little bit of yield protection there. The letters are wrong, but and Damon, did you have any?
Yeah, I think the one caveat I'll say about Wisconsin, you're probably looking at this wondering why it was inconsistent between the 2 locations. My location was an irrigated environment. So again, I think we had some adhesion issues, and we're talking about how to get around some of that this year with irrigation timing, and also, considering some of these surfactant ideas. So, that could explain, especially with the copper containing compounds, why there's an inconsistent reaction here. So keep that in mind. There's challenges here, and we're trying to layer in. We've sourced what we think is a partially resistant hybrid. So we're going to be able to look at that hybrid component in here as well as we move forward in 2022. And I think that's going to be one place to look, because we do see on the conventional side, if we choose a really good resistant hybrid, they don't respond very well to fungicides, which is a good thing in my opinion, right? So if we can carry the load with the varietal hybrid resistance, then we sort of eliminate the need to have to make a second pass, with fungicides and that sort of thing.
Okay. And I think actually, so for my letters are wrong, but I believe Badge and Oxidate were similar to the Headline, providing, and maybe Actinovate as well, a little yield boost there. Which I was surprised at some of those and how they turned out. But again, this is 1 year of data. I want to see multiple years to see consistency. But there may be some options of things that may provide a little more protection and get us to some of that yield protection that we're looking for. And I think yeah, so when we look at tar spot severity, and Damon and I probably could spend 3 hours talking about what we've been studying. We did do some work on some residue, and I didn't present that data where we see if we can reduce residue, we can layer in a little bit from the previous year of reducing that disease severity. Again, moderate resistance is what we're pushing. If we can find those hybrids that have some moderate resistance, I can see definite differences in how some of those moderately resistant hybrids can slow down that infection. We do know that conventional fungicides can increase our protection, but timing is going to be critical. In Indiana, we're still falling in that BT to R2 window is kind of my optimum window if we got to pick a time. And it's probably going to be the same with the organic products. But again, it may shift. And so this one season was the one season where we probably could have gone earlier and maybe made double application of fungicide to get a wider window of protection. But I haven't seen that consistent from every other year that we've studied it. But again, year to year on this disease, whether it's going to be a problem, and just it's important to understand if you've had a problem with your field, if you've had this history of this disease, just to be aware of it. And I didn't present the Tar Spotter app. I missed that, Damon. We do have the Tar Spotter app that's available that'll help predict whether the environmental conditions were conducive. I think I have it at the end. So that's another app to predict on it.
I think this is the other thing too with tar spot, especially. Really all the diseases is that regardless of whether it's an organic fungicide or a conventional fungicide, we need to get them on preventatively for this disease, or at least as close to the start of the epidemic as possible. Once tar spots moved into the upper canopy, it's higher than say 5% on a leaf. You're way behind the 8-ball and you're not going to catch up. Even our best fungicides out there that we know have great efficacy, if we get the timing wrong, game over. So, that's really something to keep an eye on. And we do have that tar spot prediction tool. I just dropped the link over there. We have it available for both smartphone platforms. That's free. It was developed with funding from the Wisconsin Corn Promotion Board, as well as the National Corn Growers Association. So, check that out. It at least is a tool there to help you, understand when to get out and scout and those sorts of things to try to make some of those in-season management decisions.
Okay. And so we can come back if we have more questions on tar spot as well. Damon, I'm going to throw the ball to you and you can talk about white mold since I've talked a lot.
Well, if I started in a coughing fit, I apologize. I'm fighting a cold my kids brought home. So I'll mute and get that over with, and Darcy can pinch hit for me if that happens. But one thing that we've been talking about are these diseases that are polycyclic, having multiple cycles per season. And that's complicated because if you look at a disease like tar spot, you have a long window, right, of opportunity for the fungus. And that's different when we start to talk about white mold. The opportunity for the fungus is much narrower, which in some respects makes our job a little easier, especially from an in-season management standpoint, because really what's going on here is that the formation of these little cup mushrooms here. So we've got these survival structures called sclerotia. They sort of look like rat turds. Those things are in the upper soil profile. And then as the canopy of the soybean crop closes, that is a conducive environment for the formation of these apothecia, these cupped mushrooms. And on those cups, we have lots of spores, right? And those spores are forcibly ejected up into the canopy. And so the fungus in soybeans, the fungus has to infect through these flowers. It's like 95% or more of the infections in soybean happen through the flower nodes. That's different from other crops. So if if you do dry beans and sunflowers and some of those things, this is different. We're talking about soybeans here. But, that means that if we're going to use a fungicide, this is where we're going to be targeting it, is right in that bloom, start of bloom period to the first part of pod formation. The other thing to consider here too is that, we've seen recently folks trying to push maturity group in soybeans. And when you start to push maturity group, because we have a lot of indeterminate soybean varieties grown, especially in the north central region and northeastern region of the US, that makes the bloom time quite a bit longer, right? So there's some simple things we can do by just growing shorter maturity groups, maybe giving up a little bit on the yield side. But if we got a heavy white mold field, I think pivoting to something with a little shorter maturity group can help bring that window of opportunity in a little bit for the fungus, right? We're going to shorten that bloom time. And so these are just some things to think about in addition to some of the other cultural practices. But it's really important to consider that, because once this bloom time is over, it's a couple, 3 weeks before we start to actually see the damage. When I got started at Wisconsin, everybody wanted to wait till they saw, the damage and then go out and try to do something. And by then, game over. All right. So again, it's sort of a predictive type strategy here, and then understanding how and when that bloom time happens and what the weather is like when those flowers are open. Next slide, Darcy. So, the symptoms here, it obviously gets its name because it has all this profuse white growth that you can see there in the middle of the image here. And, when it gets really bad in a field, you can see all those dead stems, on both of the side images there. We're trying to keep damage when it comes to yield loss. We're trying to keep damage down below, say, 5% or so. So 5% incidence. It's hard to pick up yield loss on a yield monitor below that level. So you can have low levels of white mold, and not see significant yield loss. The thing to keep in mind, though, is where you do have white molds, you're contributing inoculum. So in other words, those little rat turds, they're going to go back onto that soil. So any white mold is contributing back to the inoculum density that you're going to see in future years. So keep that in mind. There's some real benefits to control this disease, even if you're not detecting significant yield loss. Yeah, next slide. So we've been looking at, the roller crimping system and then trying to layer resistance, and then also, potential fungicide, Homry approved fungicide programs on top. There was some really nice work. Sarah Pethybridge out at Cornell did a really nice set of studies and published that, looking at the roller crimping system. And there's some really interesting biology that happens there. But I'll just jump to the Cliff Notes version and say that, in the roller crimping system, if you can get it right, you virtually eliminate all formation of those apothecia. Right. So it's pretty dramatic. And obviously having that thick mat laid over the surface of the soil forms a physical barrier. And then Sarah also showed that it actually stops the formation of those mushrooms. So that there's actually a change in light regimes that messes up the biology of the fungus. And so, we've been trying to dig in and look at this system and compare that with resistant varieties. I have a small breeding program for soybeans in under my umbrella here at Wisconsin. And one of the things we're trying to do is push varieties for the organic side and also the food grade market. So our main goal is trying to impart good white mold resistance. So there are public breeding efforts out there, even on the soybean side, for some of these things. So you can see the various fungicide programs, which we've layered on top here. We've got Endura at R2, which is going to be our conventional fungicide just to compare to. And then we've got double nickel, Serafol, Actinovate, and Botriostop. And we've got that repeated on both varieties. Dane is a variety out of my program, our first release, and then Minnesota 1410. So these two are going to be in the 1.4 to 1.5 maturity group range. So pretty comparable. Next slide, Darcy. So one of the things you can, kind of see here is that we didn't have a ton of white mold pressure in 2021. It was pretty hot. And so we're going to just kind of take a look at the yield data for right now and just kind of look at this in terms of, how do we start to dial things in? And one of the challenges we do have is that we need to look at the varieties and how they come through the roller crimping system. And I'm sure lots of you have a lot more experience than maybe I do in this roller crimping system. But that is one issue, is that we do see a lot of variety differences. And you can see it here between Minnesota 1410 and Dane, where, we get some yield drag. And I'll say the yield drag here in Wisconsin was so significant, we didn't even harvest the trial because, Emergence was pretty rough for us in the particular location. And so, I think when it comes to this, trying to pair that right variety with some good partial resistance and that fits into that roller crimping system is going to be the place to go. Fungicides, you can kind of see not a lot of significant differences in yield there. And again, that relates to the fact that we weren't really controlling a disease. We didn't have a lot of white mold out there. And so these things are meant to target those pathogens. And so in years where you don't have much, in terms of pathogen, you're not going to see a lot of responses there. And you can kind of see it there, even in Dura, which is our conventional standard there, not giving us much yield advantage, even over, the non-treated. I don't know, Darcy, if you have anything to add on those, what you saw in Indiana.
No, well, as I said, we had very little disease. And so, some years we have different, we try everything right. We want disease to look at them, but it's just not conducive. And so, Damon, there is a question here on spring planted rye mess up the white mold production. And I think in my mind, if we're still laying it flat and putting that mat down, I think that's where we're inhibiting that. And that's what we're trying to look at is, can we see this in soybeans as well as what they see in snap beans up in New York?
The spring rye system, typically the residue, it's grown more like an interseeded cover crop. So that the soybean or the rye only grows about 6 to 8 inches tall. So you still get more sunlight reaching the soil surface. I don't know how much it would need to totally think of a mat or how much sunlight would need to be eliminated.
Yeah, there's two really good, actually, in my mind, linked questions here. So we've been actually looking at rye stubble, spring planted rye stubble, and some other things with Rodrigo Worley, our wheat scientist here at UW-Madison. And so the reason why I'm going to weave in the row width thing right now, that question, is because the wider the rows, the more we can delay onset of white mold, right? The trade-off, though, and we've looked at this, and I laid one of the links in there on some of our recent work, is that we get a tremendous yield drag when we go to the wide rows, and there's significant weed pressure issues that we have to take into account. So, where we've sort of headed is, all right, if we're going to go in a wider row, can we put some sort of cover crop in there, or companion crops such as a spring planted rye, have that stubble there that can sort of mess up the spore flights? And we think that there is something to that. We need to do a little more work and try to dig in a little harder on that. But I think anytime you can put some sort of obstruction, physical barrier in there, you're going to really mess things up for the fungus, right? So the little cup mushrooms are very, very tiny, about the size of a number 2 pencil eraser, and they're usually really close to the ground, right? So you don't need that much of a crop canopy to really start to mess up those spore flights. So anything you can do to kind of mess with that, I think, is really, really helpful. But I also think trying to work in these 15-inch rows and then look at planting population. So to kind of get back to the row width question there, what we're seeing working quite well is that if we can go to that narrower row spacing, we can balance the weed pressure issues and kind of cut down on that, and then drop the planting populations in row. And so that's where we can sort of balance both weed control and white mold is in a system like that. The issue there, though, is that again, I think you're going to have to really dial the variety in for, if you're going to pivot to, say, a roller crimping rye system or something like that, you got to make sure you got a variety that kind of fits that system and can get through that dense canopy. So that is one caveat to keep in mind. I don't know, Darcy, you got anything else to say there on that? Or we can go to the next slide. So, yeah.
And so my other thing, and so I have it here on this bullet point, we talk about roller crimping rye varieties. I didn't have in this experiment, but actually I did a lot of work with white mold and peanuts on this. There is a biocontrol available too that's organically certified, Contans WG, and it's designed to go after the sclerotia. And so this aspect, it will break down the sclerotia, but it's to understand this one, though, if we're going to incorporate that into the system, the idea is of reducing that inoculum load. So we don't want to keep putting inoculum in, but also understand it's got to have time to feed on the sclerotia. And so some of these products may be it's not a short-term answer, it's a long-term plan, and it may take a couple years. If you wanted to incorporate, if you have a hot spot, if I was looking at a field and I had a hot spot of white mold, I'd be throwing in probably some Contans to reduce that sclerotia, and then layering in these other things as well to try to minimize the amount of disease and try to tackle the disease at the various aspects of its life cycle. So if we tackle, this one, we can tackle the inoculum load at the beginning, and then trying to manage the protection in the crop canopy in season as well.
Yeah, we've had some growers here in Wisconsin, especially in the central sands under center pivots. They've been using contans under those pivots, against white mold. Had pretty good luck. I think, if you talk to those farmers though, and I've seen this as well, you have to be patient. So that's one thing. And Darcy kind of mentioned that. And it may take a couple applications actually to build up, that mycoparasite in the soil. The other thing is you have to sort of adjust a little bit to what the label says. The last label I looked at, it was like it was a pre-plant type application. And Mike Wunsch out at North Dakota has been doing some of this work in dry beans, and he showed some nice responses where they actually apply it in the fall, and you make sure you got some moisture there. And that basically gives the mycoparasite a head start, right, before the next crop is there. And so, some simple adjustments like that, and then making sure you can pencil it out, the cost out across your rotation, I think are important considerations if you want to pivot there. But I do think that that's a really viable option to get rid of those initial inoculum propagules.
Yep. I was saying peanuts as well. We were putting a fall application out multiple years where it worked on reducing that. But it's just understanding when we start incorporating these biologicals, whatever it is in any system, you have to understand how they're interacting, and they have to be in touch with the pathogen in order to have activity on that, or they have to be there and protecting or, competing on that leaf surface to not allow the pathogen in. And so each biological is going to have its different mode of action, which is different from what we're dealing on the fungicide side of things. I'm trying to think what else I have left on the slides. So some other things just to so you guys have this is the tools that we do have for these 3 diseases that help predict, are the conditions conducive for these diseases in season. So again, that website with the Scab Smart, that is active right now for monitoring as wheat starts greening up across the country. Damon Sporecaster is available for the white mold, and then you can put into that, I believe, the growth stages as well as whether you're at the high risk. And then we have the Tar Spotter app too as well. And I think, utilize these tools if you have problems on your farm. They're just great tools to give you an idea of, is the risk high? Do I need to think about something else to try to protect against the diseases in addition to what else the other things that you're working on? And again, some other tools. Again, I think it's important. You need to know the problems are in the field. So first and foremost, what are the problems there? Do you have proper disease ID? If you're not sure, send them into our clinic and they'll help confirm it. We both have clinics, both in Wisconsin, in Indiana. Every state probably has some disease diagnostic lab that can help. We are tracking various diseases. So like tar spot is one, we have a national map that we track. It'll give you an idea of when I find it active in Indiana, I'm going to turn those counties yellow to say, yes, it's moving and active in the crop canopy. As we saw in this last season, we were 2 weeks earlier in 2021 than we were in 2019 and 2020. And so we were already kind of hedging the bet that the disease started earlier and it was going to lead to some significant losses in northern Indiana, at least. But there's a lot of tools out there to help try to predict diseases. I know diseases aren't the only thing we're trying to manage on our farms, but we're just trying to give you some tools that if the disease becomes an issue in a certain field, there are some of these tools available. So then Damon, I'll go back. I think he shared some of the crop protection network. So this is where we share some of our multi-state data. So there's a lot of resources available there. There's a new white mold, what do we call it, web book available. There's a tar spot web book available where we try to incorporate some of the information that we're learning as we learn more about each of these different pathogen systems.
Yeah, I've shared all the links a couple of times over in the chat there. So you can grab those. And I think Aaron will send those out.
It should all be saved in the archive chat, and I'll send that out to everyone in case you haven't had a chance to cut and paste those so that everyone has access to those. And I'll give Aaron I'll end the slideshow now, but I'll send her a PDF of that too, so you can have all the slides there that we've shared on some of that information as well. So feel free to reach out if you have thoughts or ideas. We're trying to evolve and try to work more in organic disease management, and just try to give you some of the tools that we've built on the conventional side of things as well. That's great. It's great to see some of these biologicals start to be looked at a bit more closely. Does anyone have any additional questions on the diseases that have been discussed so far, or any other questions more generally? Any other people have experience with any of these compounds that have been discussed?
any other, or I guess on the disease side of things, because that's what we deal with. Are there any other issues that you've run across on your farm that you're having problems in your systems?
One question I have is, have you ever worked with Bavaria bassiana as a seed treatment?
Not.
I have not. Nope, I have not. That is a major area though of concern for me, especially in the rye roller crimp system, is how do we get, how do we balance seed rot issues, right, in the organic system? So there's an added challenge there. We sort of deal with that on the conventional side by just putting conventional fungicide treatments on, right? And so, that's certainly an issue. And then if we're dropping populations down a little bit to try to deal with white mold, there's an added concern there. So I think we need to dig into those a little further for sure to see how we manage, the early blight, early seedling blight problems and that sort of thing for sure.
Tried any of the chitosan type products at all as an adjuvant?
Nope. I'll take all ideas though.
Yep. Feel free to send us stuff.
Maybe I can share it with Aaron. We did do some foliars this year on our barley. We pulled a sample from each field that we sprayed, and the treated fields it was a combination of the chitosan product and fish and like a biological and some micros, but the fields that we did spray were undetectable to or half of what we were without. So there was a response.
Oh, wow.
So it was like from 0.8 on the untreated, and then 0.5 to 0.33 undetectable range where it was treated. And we had a lot of
sharing that, Bob. It'd be great if you could send some more information. That would be super.
And when we were combining, I thought it was going to be a total disaster because there was nothing but black dust. So it was on the stems and on the leaves, but it was not infected in the kernel.
Yeah, last year was a bad problem for sooty molds that moved in late in the season because we were really dry, and a lot of, especially, I don't know where you're located, but especially here in the Midwest, we were so dry that we had a lot of insect feeding, which made honeydew. And we got tons of, sooty mold basically on the outside of those kernels and a lot of the small grains crops I looked at. And actually, for the first time in my career, I actually saw black point, which is when those sooty mold fungi actually make it all the way into the kernel and actually start to damage the kernel. And that's the first time I've ever seen it in the 15 years I've been doing this. So, it was an interesting year last year for sure.
Yeah. It still was a tough year because it caused just enough staining and discoloration that it caused a flavor issue.
Yeah. So my comment on trying those products is just leave a check strip or two to make sure you're seeing differences and what's the benefit. That's some of those things if you're looking at new products. Or you said you had 2 fields to compare. So that's what I try to tell my guys. If you're going to try something new, leave some check strips so you really can see if there was an added benefit of putting something out there.
Yeah, and I'm writing down all these things you're dropping into the chat, so keep sharing away. One of the things that we do in the Crop Protection Network is we put out coordinated trials, and this has been our goal with the SARE project, is to get a coordinated project started for organic products, so we can start generating similar efficacy data like we do for a lot of the conventional fungicides. So, we're not locked into just the things we're looking at. That was just a place to start. So I'm loving all these comments, and I've got a list going here of what everybody's mentioning.
Yeah, if you look at it, there's over 200 plus biologicals labeled or that hit EPA approval. So it's trying to figure out those ones that might work, right? And so I pulled from my vegetable work back in the day before I started at Purdue and some of those things that I was seeing some activity in some of our organic vegetables. So that's kind of where we're trying to hone in on what products to start with, and then where do we go from now? So we're always looking for feedback to feed into this project, and that's just the hope, is to start something to keep moving forward with it.
Are there questions or comments? All right. Well, let's thank Darcy and Damon for all the information, and thank you. So this is such a needed area. I'm just so excited that there's more of a infrastructure and effort that's being devoted to this, to do more systematic evaluation of these products and understand where they're useful, and where we might be able to start being able to make more sound recommendations to organic grain farmers. So I'm excited to hear more from you both as the project develops. So have a great weekend, everybody, and 2 more workshops, one on organic markets next week, and then follow that organic crop insurance. And we have a full slate of field days this summer. I'm so excited about how our field days are shaping up. So we'll hopefully be seeing some of y'all in person as we get to our summer programming. So we'll see you all soon. Have a great weekend. Bye, everybody.
Thank you.
Thank you.